In this video, we're going to look in depth at transcription. Remember, transcription is happening in the nucleus, and it is where DNA is being turned into code on mRNA, or messenger RNA. So we're going to need to look at taking our DNA strand and uncoiling it from its double helix structure so that the gene is exposed and can be copied. Now we need to think of a little bit of terminology here. We have a coding strand that has the code for the protein on it. That is our gene. We have the opposite strand of the DNA that is called the template strand. You're aware that we have bases in DNA, so let's make up a sequence of bases on the coding strand comprised of the 4 bases that are found in DNA. So I've got a sequence here, A, T, G, C, A, C, and that means on the template strand we will have the base pairs for that. So opposite the A will be a T, opposite the T will be an A, opposite the G will be a C, a G, a T, and another G. So this is just a piece of DNA that we have pulled apart from its double helix. Now an enzyme comes in called RNA polymerase, and it will start to match up RNA nucleotides base pairing with the DNA. So it's completely dictated by the code found in the DNA. So opposite the T there will be an A. Opposite the A you would expect to find a T, but in RNA there is no thymine. It's replaced by a base called uracil. Opposite the C there will be a G, and then the enzyme will place a C here, an A and a C here. So the RNA is built according to the code that you find in the DNA, the only difference being that where you would want to put a T, you put a U for uracil. And the RNA continues to be built. There's a long line, and eventually will peel off the gene when it gets to the end of the gene, and then that piece of mRNA can then leave through the nuclear pores and go off to the ribosome. So we've got some interesting things happening here. We have the bases in DNA, so A, T, C and G in DNA, matching up complementary base pairing with the bases in mRNA. Remember, you would want to put a T here, but you have U instead. All the other bases base pair as you would expect. And the enzyme involved in making mRNA is called RNA polymerase. Because it is an enzyme, so it ends in A's, that makes a polymer, so a molecule made of many small pieces of RNA. So an RNA polymerase is doing all of this. It's doing the finding where the start of the gene is, opening up the DNA helix, copying the template of the coding strand, and making complementary base pair copy so that we carry the integrity of the original message out to the cytoplasm. That is transcription.